Hi everyone, welcome back. So last week we took a look at how chemicals are bonding together to form ionic bonds and covalent bonds and hydrogen bonds. And we also looked at the structure of water. So today we're going to apply what we learned last week into exploring macromolecules. So we're going to, and basically this covers the information from chapter 3. So, like always, I highly suggest that you take notes using the course objectives. We'll probably get through 3.1, 3.2, and 3.3 today. We might get into lipids as well. I usually try to keep these presentations around 100 or an hour and 20 minutes. Okay, so let me go ahead and get this started. Oops, wrong way. Okay, so let's get into chapter 3. So taking that information we learned last week and applying it to our macromolecules of the body. So we're going to take a look at basically 5 themes today. So we're going to start out by discussing molecular organization, particularly with our carbon-based molecules. And then we're going to get into the 4 macromolecules of the body, starting with carbohydrates and lipids. And then and then that'll probably cover this chapter, and then, or this lecture. And then the next lecture, I'll get into proteins and nucleic acids. Okay. So let's start out by talking about what basically what carbon compounds are and why they are so vital for life. So living things are made up of chemicals that are mostly based on the element of carbon. So you are carbon based and you are made of molecules that are carbon based. So carbon, the reason why carbon is so vital for life is because it has 4 valence electrons in its outermost shell. Now, last time we talked about the valence shell where those electrons are. And because it has 4 valence electrons, it has an ability to form a lot of complex molecules and varied molecules because in order for it to fill its outermost shell, it has to gain 4 electrons or lose 4 electrons. So that's makes it a very versatile molecule. And anything that contains a compound that contains carbon is what we classify as an organic compound. So things like our carbohydrates, those are organic compounds because they, or organic molecules because they contain carbon or they're carbon based. And the ones that we're going to study for this class, because they are basically in living things, are carbohydrates, lipids, proteins, and nucleic acids. And these are what we call macromolecules, so macro being large, so the very big molecules. Okay. So why is carbon so versatile? So we talked about already how it has 4 valence electrons in its outermost shell. And because of that, it doesn't really form ionic bonds because then it would have to lose 4 or gain 4 electrons. Probably not going to happen. So it's going to form covalent bonds. And so it can, because it has 4 empty spots in its valence shell, it can have 4 covalent bonds, bonding with a variety of different atoms. And that allows them to form very large and complex molecules. Molecules with multiple carbons bonded together, they usually form kind of a tetrahedral shape, so you can get some unique shapes to our carbon-based molecules. And then when we have 2 carbons joined together, they can form a double bond. And the atoms joined in the carbons are in the same plane as the carbons. So we'll talk about how the carbon's versatility and this complex shapes that they can form and so forth. Okay, so let me give you an idea of the different shapes. So here are some different shapes you can see in the table here. So something like methane, is basically just one carbon atom covalently bonded to 4 different hydrogen atoms. And so this configuration forms kind of this like tetrahedral shape. Ethane is a molecule that has 2 carbons, so they'll bond with each other and then they have the 3 hydrogens each. And so you can see how they are situated where the carbons are kind of on a horizontal plane, and then the hydrogens are surrounding them. Okay, and ethylene, which is very similar to ethane, where you can see how ethane has 2 carbons, and so does ethene, but ethene has a double bond between the 2 carbons, and so that allows them to only need 4 hydrogens instead of 6. And so you can see how it creates this flat plane. Okay. So there's different shapes. Just by, we're basically just working with 2 atoms here, carbon and hydrogens, and you can see the unique structures that are being formed with just those 2 elements. Okay, so let's talk about how carbon forms its covalent bonds. So the number of bonds an atom can form is in its valence. So we talked about this last week. So the electron configuration is based on how many electrons are in their outermost shell or their valence shell. So we talked about how hydrogen has 1 electron in its valence shell, so it wants to acquire another one to fill its shell. Oxygen, it has 6 in its outermost shell, so it wants to gain 2, so it has a valence of 2. Nitrogen, it has 5 in its outermost shell, so it wants to gain 3, so its valence is 3. Carbon has 4 in its valence shell, so it wants to gain 4. So because of this need to, it cannot exactly lose 4 or gain 4, it's going to bond covalently with up to 4 different atoms. Okay, so it's compatible with many different elements. So let's take a look at something like carbon dioxide. Carbon atoms can partner with atoms other than hydrogen. So for example, we have carbon dioxide. In carbon dioxide, carbon has, what's their valence again? It was 4, right? And so you can notice how this carbon atom is in the center here of the structure, and it has a double bond with 2 oxygen atoms to form that structure. Okay, so it can form other bonds aside from forming with hydrogen. And you can see that in this case, this was a double bond. Okay, and then here's another example. We have a carbon-based molecule, an organic compound, where we have 3 carbons, and they have a single bond each, and then they are bonded to the hydrogens. Okay. So let's take a closer look at what we call carbon skeletons, where we look at how the carbons are arranged.
The carbon backbones of organic molecules may vary in length, as shown here by a carbon molecule with 2 carbons on the left and another molecule with 4 carbons. Carbon skeletons may have single bonds or double bonds, which can vary in location, resulting in molecules with different properties. Carbon skeletons may be unbranched or branched, and some carbon skeletons are arranged in rings. Such variation in carbon skeletons is one important source of the molecular complexity and diversity that characterize living matter.
Okay, so from that video we see that there's a variety in the carbon skeletons. So we're going to go through that variety in a little bit more detail. Okay, so carbon atoms, they form these chains. So you have carbon, 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 and we call those chains skeletons. And they vary in length. So we could have, here's an example of a carbon chain where there's just 2 carbon atoms. Here's one where there's 3 carbon atoms. Okay, so they form these chains. Another thing that's interesting about the carbon skeleton is that they have abilities to branch. So you can see how in this case we have 4 carbons lined up in a linear plane, but then here we have 3 carbons lined up, and then the 4th carbon is branching off of the middle one. Okay, so carbon chains can be branched or unbranched. Double bond positions. So here we have two molecules of butene, and they actually have the same number of carbon atoms and same number of hydrogen atoms. But look at the difference in the position of the double bond. So the carbon skeleton is lined up essentially the same, with the exception of the double bond, and so that's going to change the structure. Okay, and then finally the last one, the presence of rings. So carbon can form these little carbon rings. So in this case, when we do see a carbon ring, the carbons can be either single bonded to each other or they could be double bonded. And so you can see how when we draw it out, how we draw the double bond versus the single bond. And then we can actually even take away the hydrogens and the carbon labeling and just have a little hexagon. So that's a single bonded carbon hexagon, cyclohexane, or a double bonded carbon skeleton hexagon called benzene. All right, so there's a couple of ways we can draw it. We can leave out those hydrogens if we want. All right, so we talked about how there's a versatility in the structure of the carbon atoms, how they could form these different varying carbon skeletons. And sometimes, like I just mentioned in the last slide, we can have a situation where we have 2 molecules that have the same molecular formula. They have the same number of carbons, the same number of whatever else, but they have a slightly different structure. So when that happens, we call them isomers. So isomers have the same molecular formula, but different structures and properties. So for example, let's take a look at pentane here. So how many carbons is there? There's 5, right? So we would write the molecular formula as C5. How many hydrogens do we see? So there's 5, 10, 12. So the molecular formula is C5H12. If we look at 2-methylbutane, there's 5 carbons. And if I count all these hydrogens, 3, 6, 10, oh, there's 12 as well. So notice they have the same exact molecular formulas. But in this case, we have a linear setup, and then we have basically an unbranched versus a branched. So this is an example of a structural isomer. So you can see that the bonds are slightly different arrangement, okay? There's other types of isomers, so this is considered a structural because you got like the unbranched versus the branched. It differs in the covalent arrangement of the atoms. We can also have something called a cis-trans isomer. So in cis-trans isomers, you'll notice the position of the hydrogens in relation to the other isomer. So in this case, in this made up molecule, we have carbons double bonded with each other. And then you notice we have the hydrogens on one side. Okay, so this is called a cis isomer because the other ones are on one side. Okay, so cis, think same side. Trans means opposite. So instead of the hydrogens on one side, they're on opposite sides. And so that means that whatever other bond it has is also going to be on the opposite side. So that would be a trans isomer. So cis, think same, trans, think opposite. And so carbons, they still have the same molecular formula, so they're isomers of each other, but in this case, the atoms, they differ in their spatial arrangement. So you have either the same side for the cis or the opposite side for the trans. Okay. And then finally, the last type of isomer we're going to talk about is an enantiomer. So enantiomers, those they have a unique structure where they're mirror images of each other. So they tend to be asymmetrical. So you can see how we have these two molecules, the L isomer and the D isomer. And they have, it looks like the same shape. They have a tetrahedral shape, but the arrangement is slightly different. So they're actually mirror images of each other. Okay, so enantiomers, think mirror images. So little slight differences. Okay. So think of it as like the left hand and the right hand version of a molecule. All right. So here's our first checkpoint question of the lecture. So one isomer of methamphetamine is the addictive illegal drug known as crank. The other is a medicine for sinus congestion, which I desperately need right now. So how can you explain the differing effects of the two isomers? Well, remember that the structure determines the function. So if they are slightly different structurally, they're going to function differently. So structure and function work together. So isomers, they have different structures, and so that's going to have them cause different functions in the body. All right? Okay, so now that we have a little bit of understanding of our friend, the carbon atom, we're going to see how we can end up having these complex macromolecules form, and how we can use certain chemical groups that we notice occur often within these macromolecules, how we can use those groups to identify the function of that particular type of molecule. So chemical groups, they can replace one or more of the hydrogens bonded to that carbon skeleton of a hydrocarbon. So we call those functional groups. So they're chemical groups that affect the function of the molecule by being directly involved in the chemical reactions. So each functional group actually has its own chemical characteristic. So for example, look at down here, we have two hormones. These are the two sex hormones, estradiol and testosterone. Now, if you kind of just look at it briefly, it looks very much the same, okay? So the carbon skeleton is actually the same, pretty much. I mean, you see that there's these little hexagons. Here's a pentagon, hexagon. Oh, here you have a double bond on one and only one double bond on the other. But then notice you have these things in blue. So these are called functional groups, and the functional groups are going to affect the function of that hormone. So even though the structure is very, very similar to each other, the presence or absence of certain functional groups tremendously impacts the way that particular molecule functions in the body. So we're going to go through the different functional groups. And so you'll need to know these functional groups and understand, give some examples and understand what they do, okay? So this will help us, especially when we get into the macromolecules. So here's the first four. There's 7 of them total. So the first one we're going to talk about is called the hydroxyl group. So we talked about the hydroxyl ions last time with the properties of waters. Remember, the presence of these guys in great quantities make more of a basic situation, or basic, sorry, I got a runny nose. So it creates more of a basic solution there with the presence of high amounts of the hydroxyl ions. Well, we can also have something called a hydroxyl group. So that's where we have the oxygen and hydrogen bonded to the carbon, okay? And we typically call these type of molecules alcohols if they are present as that functional group in the carbon chain. So here's an example. You have a carbon chain, here's the hydrogens around, and then you have this extra hydroxyl group. So the hydroxyl group is replacing one of those hydrogens, and so that would be considered a functional group. And so now we have a type of molecule that's called ethanol. So this is a hydrocarbon. There's lots of hydrogen, but it actually has an OH group that changes its function slightly. Okay. The next functional group we're going to talk about is called a carbonyl group. So carbonyl groups, we have the carbon is double bonded to an oxygen atom. So you can see that double bond there. And these molecules that have these functional groups are usually known as ketones or aldehydes. So here's a couple examples of ketones and aldehydes. So acetone. Acetone is a 3-carbon chain. You notice the hydrogens, but then there's a double bond to an oxygen atom. Okay, so this is an example of a functional group that has a carbonyl group attached. Propanol has also a double bond carbon, so it has that carbonyl group. Okay. So we have hydroxyl, carbonyl, and then let's go ahead and put them together to form a carboxyl group. So think of carboxyl as carbonyl plus hydroxyl together, because that's essentially what it is. You have the carbon attached to a hydroxyl group and a carbonyl group. So our molecules that have a carboxyl group, they're known as carboxylic acids or organic acids. So think of like acetic acid, citric acid, things like that. They all have this carboxyl group attached. Here's an ionized form. So these, since they're acids, they're going to, of course, be more likely to release that hydrogen into the solution to make it the pH go down. Okay, so carboxyl groups, you can kind of guess if they're going to maybe perhaps change the pH of a solution if they dissolve in there. Okay, amino groups have nitrogen. So the carbon is going to be attached to a nitrogen with 2 hydrogens. So we call those amines. So they're important, especially when we get into proteins. So our amino acids all have these amino groups. That's why they're called amino acids. And ironically, amino acids also have, what's that guy over there? A carboxyl group. So that's why it's called an amino acid, because it has an amino group on one end and a carboxyl group on the other. So now you're starting to get it, right? So we got, that's why we have these names. So here is glycine. This is a type of amino acid, and here's the amino group, and then there's that carboxyl group to make it an acid. So here's an example. And because of the carboxyl group, it has the ability to ionize, and then to release that extra so we end up with that ability to dissolve in the water and so forth. So that's the amine group. Okay. Moving on to the sulfhydryl group. So sulfur, hydrogen together form a sulfhydryl group. Pretty easy. Okay, so you have a sulfur atom and a hydrogen atom bonded to the carbon. That would be known as a thiol. And here's an example. If you take a look at cysteine here, so cysteine has this sulfhydryl group. Notice any other functional groups there? Hey, what's that? Hey, what's that? Okay, so we're starting to identify. This is actually an amino acid. Cysteine is amino acid because it has an amino group and an carboxylic acid, carboxyl group. But it also has this sulfhydryl group, so that unique thiol compound is going to be added. Phosphate groups have phosphorus, so you have the P surrounded by oxygen. So it's going to be basically a double bond to oxygen and then 3 bonds to surrounding the rest. And that will be attached to some carbon skeleton. So here's a glycerol phosphate. So you can see the carbon skeleton, and then you can see how that oxygen is going to be single bonded to the carbon. And then we have the double bond for the phosphorus, and then we also have the other oxygen. And in this case, notice the electronegative symbol. So it's going to be a little bit more electronegative. So glycerol phosphate has kind of a negative end to it. And then the final group is the methyl group. So methyl is basically one carbon with 3 hydrogens, and then we attach that to something else, another carbon chain. So we call that a methylated compound. So here's an example. Here we have, this is not a carbon ring because there's nitrogen in there, but you can see how that carbon, we have this methyl group that's attached. Okay. So this is 5-methylcysteine, or cytosine, excuse me. And you can see some of the other things that are there. All right. So we went through the 7 different functional groups. So make sure you can identify them because that's going to help you understand which molecule we're talking about. And remember, the functional group can cause big changes in the form and function of the body. So like I said before, testosterone and estradiol, very similar hormones, okay, in their structure, but the functional groups are making them very different from each other. So what functional group was this? Okay, so a carbon and 3 hydrogens attached to the carbon chain. So that was a methyl, okay, methyl group. What was the double bond to the oxygen? That's a carbonyl group. Okay, so testosterone has a methyl group and a carbonyl group attached to the hormone structure, whereas estrogen only has that hydroxyl group. It doesn't have the other two. And because just those slight changes in functional groups, you get big changes in the way they function in the body, right? Testosterone is more what you think of as the male hormone, though females produce it as well. But you can see the difference in the function inside, for example, the lions. So lions, male lions, they have a lot of testosterone, so that allows them to grow their big mane, and their body structure is a lot bigger, and their head's a lot bigger, and they're more aggressive and muscular. The female doesn't have that because she's got more estradiol. Okay. All right, so we talked about the functional groups. Make sure you understand those. And now we're going to see how molecules form and break down. So chemicals and cells, they make different molecules and they break them down. And it goes through these same processes. So basically, we call a polymer is some sort of molecule, a larger form of a molecule that can be broken down into smaller forms. And there's 2 processes that are involved in order for a molecule to be broken down. And then another process for it to build up into a bigger molecule. So the first one we're going to talk about is dehydration reactions. So dehydration reaction is basically where we're going to build a bigger molecule from smaller subunits. And those smaller subunits are called monomers. So a monomer is a subunit. So when we have 2 subunits, we can bring them together to form a polymer. So think monomer, mono means one, polymer, poly means many. So one subunit, if we have 2 subunits, we want to bring them together and form a bond, we need a dehydration reaction to occur. And the way this occurs is, well, think of this term, dehydration. Well, what happens when you're dehydrated? You've lost water, right? Well, that reaction is losing water. So it's basically, it takes these 2 monomer subunits and brings it together. But in order to bring them together, we have to remove a water molecule in the process. And I'll show you how that's done in a second. So think dehydration, we're removing water to stick 2 monomers together. Polymers, they can be broken down into monomers by the opposite type of reaction called hydrolysis. So hydrolysis, well, hydro means water, lysis means breaking. So remember how dehydration, we removed water to bring 2 molecules together? In hydrolysis, we're going to add water to break apart 2 monomers into separate subunits. So hydrolysis, we're breaking apart a polymer into monomers by adding water. Okay? Now, normally if you have, two monomers sitting next to each other, the time it takes for that reaction to occur is going to be very, very slow. Okay, it might eventually happen, but it's not. It needs a helper. So enzymes catalyze these reactions. So in order to have two monomers brought together to form a polymer for a dehydration reaction, we need to have an enzyme speed it up. Same with the hydrolysis reaction. An enzyme would speed it up as well. Okay. So let's go ahead and watch an animation showing this. Oh, I gotta put my pen away.
Small molecules called monomers are assembled into long chains called polymers by a dehydration reaction. Let's look at this reaction more closely. An enzyme covalently bonds two monomers to each other with the loss of a water molecule. One monomer provides a hydrogen, H, while the other provides a hydroxyl group, OH, and water is formed as a byproduct. This reaction is repeated as monomers are added to the chain one by one, making a polymer. Polymers are disassembled to monomers by hydrolysis. A different enzyme breaks the bond between monomers by adding a water molecule, with a hydrogen from water attaching to one monomer and the hydroxyl group, OH, attaching to the other.
Okay, so that animation not only showed us a dehydration reaction, but also a hydrolysis reaction. So let's take a closer look in a figure here. So we see this figure. The first part is a dehydration reaction. So dehydration, remember, we are dehydrating it. So we're going to remove water in order to bring two monomers together. So let's take a look at the monomer. Well, here, this isn't a monomer, it's a short polymer. It's got 3 subunits. But notice at one end of the subunit there is a hydroxyl group, and at the other end there's a hydrogen. At this separate monomer has a hydroxyl group and a hydrogen group. Okay, so as we undergo a dehydration reaction, we have to dehydrate it. We got to remove a water molecule. So in order to do that, this hydroxyl group, OH group, is going to break off along with this hydrogen group to form water. And that's going to allow the bond to occur between these two carbons, or these two monomers. It doesn't have to be carbon. Okay, so now they formed a covalent bond. All right, so we're removing water in order to form a bond. Now that's dehydration. Let's hydrolyze it. So we can do the opposite, where we can take water and add it to a reaction in order to break apart the subunits. Okay, so here we have our polymer. We're going to add the water to it, and that's going to break apart the chain. And then that way the water is broken into hydrogen and OH. Okay, so hydrolysis, we're breaking apart a polymer into subunits. Dehydration, we're removing water to bring two subunits together. All right. So here's a question for you. Suppose you eat some cheese. Cheese is good, right? So what reactions must occur for the protein of the cheese to be broken down into its amino acid monomers, and then for these monomers to be converted to proteins in your body? So let's break this question down. Okay, so let's do the first part first, and then we can get to the second part. So what reactions must occur for the protein of the cheese to be broken down into amino acids? So we have a big protein, we need to break it down. So what type of reaction do we need in order to take that large molecule and break it down into smaller subunits? So we need a hydrolysis reaction. So we need to add water to break apart the protein into smaller subunits, which we call the amino acids. Okay, so now in our body, that's what our digestive system does, is it basically is breaking down the proteins and things like that into its monomers or its small subunits so that you can absorb it into your bloodstream. Okay, there's no way, like if you ate a piece of cheese, that can fit into a cell, right? You got to break it down into small units. And so hydrolysis is going to break it down so that your body can absorb the amino acids. But then we need to make proteins from the amino acids. So how do we do that? Well, we take those amino acids, and those amino acids undergo what type of reaction to make the proteins that we need? A dehydration reaction. So we're going to remove water between the amino acids to bring them together to form proteins. Okay. All right. So now that we've had an understanding of the chemistry that's involved, we're going to start taking a closer look at the 4 major macromolecules of the body. So those are the carbohydrates, the lipids, the proteins, and the nucleic acids. And each one of these molecules is a macromolecule. So that means it has thousands of different, or possibly it can have thousands of different subunits to it. Okay. So we're going to get into, we're going to actually watch a video first. So this video is, again, an Amoeba Sisters video. And it's going to help us, basically give us a little analogy and so forth, so for us to remember like the structures and things.
Go ahead and think for a moment about your very favorite food. What is it? Pizza, macaroni and cheese, chicken salad, sushi? Well, we all have different food preferences, but food is a source of large molecules that are needed for life called biomolecules. There are 4 major biomolecules that make up all of life, and this will be the focus of this video. Before we get into details about the 4 biomolecules, we need to talk about one very important vocabulary word, the word monomer. A monomer is a building block. If I had some large substance, the parts that make up the substance are called monomers, just like building blocks. We're going to talk a lot about monomers today because we need to understand what the biomolecules are made of. And we need to understand biomolecules because they're building components of life. So let's introduce the 4 biomolecules now and talk a little bit about their functions. We'll start with carbohydrates, carbs. Well, carbs are something that you've probably heard a lot about when people are talking about diets. You know, they try to go low carb, or maybe they want a lot of carbs. Diets always come and go. Pasta and breads are examples of foods heavy in carbohydrates. Carbs are actually a very important source of energy. In fact, that's one big function of carbs. They are a great, fast source of energy. If you were a marathon runner, you might want to eat a lot of carbs the night before a race. Lots of marathon runners do this. It's called pasta loading. They eat a big pasta dinner the night before they go out on their marathon. Now, carbs have a monomer. Again, remember, monomers are building blocks. The monomer for a carb is a monosaccharide. I know that's a big mouthful, but monosaccharides make up carbohydrates. Next one up is a diverse group known as lipids. Lipids are better known as fats, and they have 2 different types of building blocks. One type of building block is called a fatty acid, and the other type is called a glycerol. Now, examples of lipids include butter, oil, and cholesterol. Lipids, though, they have a lot of great functions. You might think, well, that's fat. How good can fat be? Well, it just depends when you put it into context. For example, you know those really adorable seals that you see on calendars? They have this fluffy white hair. They're actually called a harp seal. Well, they only look like that when they're babies. When they get older, they're not quite as cute. In their little baby stage, they actually have a lot of this hair that they're born with. It keeps them warm. But over time, they have to develop blubber. It's fat, and it helps keep them warm. Lipids are great for insulating. Also, you might not think about fats as being related to energy, but fats are a great source of long-term energy. They can store energy for a long, long time. Say, for example, you wanted to swim the English Channel. That's like 21 miles of swimming. The fastest swimmers might be able to do that in 7 or 8 hours, but It might take a lot longer than that for the average swimmer, more like 25 hours, and that's a lot of swimming. Well, you would want to make sure that your body has enough lipids, enough stored fat that it can pull upon. Because after you burn off those carbohydrates, remember, carbs are the fast source of energy, well, you might not have enough energy storage unless you have some lipids on hand. Lipids also make up cell membranes. So they are very important for life because all living things are made of cells. Of course, an excessive amount of lipids could be a bad thing for your health. Remember, it's all about moderation. Okay, next, proteins. Now, when you hear about proteins, a lot of times you might think about power bars. They say they have a lot of protein in them and that they help with muscle building. Well, protein is great for muscle building. Examples of foods that are high in protein include meats and many types of beans. The monomers of protein are amino acids. So sometimes you see these labels that say, this has 20 amino acids in this food. And really, they're just trying to say that it has protein because proteins are made of amino acids, so that's just some fancy advertising for you. But in addition to being important for muscle development, protein is also very important for other functions, such as working in the immune system and acting as enzymes. Remember, enzymes are made of proteins, so proteins are important for the body. Now, when we start talking about genes, the DNA genes, not the genes you wear, the DNA codes for proteins that are very important for structure and function in the body. The last biomolecule is known as a nucleic acid. Nucleic acids include DNA and RNA, which we'll get to when we get to genetics. They have a monomer called a nucleotide. That's going to be an easy one for you to remember because nucleotide sounds a lot like nucleic acid. And if considering DNA and RNA, both of these are involved in genetic information for the coding of your traits. They are found in a lot of your food because whenever you eat something that came from something once living, it can still contain the DNA. For example, when you eat a strawberry, you're actually consuming all the cells that make up that strawberry. And in the nucleus of all those strawberry cells is DNA. Plants and animals both have DNA. In fact, any type of life must contain nucleic acids to direct the cell's activities. So we just powered through introducing the 4 biomolecules by providing examples, exploring their monomers, and giving some general functions. One last very important part to mention is the structure of these biomolecules. Understanding the structure can help with predicting their properties and easily being able to identify them. One thing I like to tell students to do is to write the 4 biomolecules in the same order that we went through: carbs, lipids, proteins, and nucleic acids. Then remember this mnemonic device that goes with these 4 biomolecules: cho, cho, chom, chomp. Instead of chomp at the end with an M, it's chomp at the end with an N. The C stands for carbon, the H stands for hydrogen, the O for oxygen. So carbs, lipids, proteins, and nucleic acids all have that CHO in there. It's just that proteins and nucleic acids also have an N, which is nitrogen. And nucleic acids additionally have a P, which is for phosphorus. So again, CHO, CHO, CHOM, CHOMP, the major elements in the 4 biomolecules. Now, these elements are arranged differently in the 4 biomolecules. It's important to explore the arrangement of the elements in biomolecules because the structure of that arrangement greatly impacts the biomolecule function. So, to the Google to discover some biomolecule arrangement illustrations. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so hopefully that gave you a good overview of the 4 macromolecules we're going to be talking about. And I liked how they used that cho, cho, chon, chomp because that's going to be helpful in us figuring out the structure of these macromolecules. So we're going to start out by learning more about carbohydrates. So remember from the video, the carbohydrates are these macromolecules that are made up of subunits, and those subunits are called monosaccharides. So when we think of a carb, we think of sugars. And so the monosaccharides are the simple sugars. So those are going to be the monomers of the carbohydrate, okay? Now the carbohydrates, the polymer is going to be the polysaccharides. So polysaccharides are a bunch of monosaccharides linked together. And an example of a polysaccharide would be starch. So this one's really easy to remember, the monomers and the polymers, because you just basically add saccharide to mean carbohydrate. So monosaccharide is the monomer. Polysaccharide is the polymer. So that should be easy. Just remember saccharide. Okay, so the structure is basically monosaccharides building into the polysaccharides, such as starch. All right, so if we take a closer look at our structure here, here's an example of a monosaccharide. And notice we have 6 carbons in a row, and they can form these little rings. And usually with our monosaccharides, these carbon rings can be made of anywhere from 3 carbons to 7 carbons. So you end up with these little ring structures. Okay, so that's something that you'll notice with our carbohydrates. They all have these ring structures within them. Okay. So this is an example of how many carbons was that? 1, 2, 3, 4, 5, 6. So this is a 6 carbon ring, okay, a hexose. So the monomer for carbohydrate is the monosaccharides. And what's interesting about the monosaccharides is that they have a very unique molecular formula that makes them up. So they are always in the same ratio of 1 carbon to 2 hydrogens to 1 oxygen. So basically, if we have a 3 carbon ring, so that would be C3H6O3. So it's always going to be in that same ratio where we have the carbons and the oxygens will be the same, equal each other, and then there's always twice as much hydrogen. So that's one thing that we'll notice, for example, with glucose. Look at glucose. So it has 6 hydrogens, same amount of oxygens, but double the amount of 6 carbons, same amount of oxygens, but it doubles the amount of hydrogens. Okay, fructose, it actually has the same symbol. Hey, what's it called when we have 2 molecules that have the same molecular formula? Was that a isomer? Okay. So it's basically what we do is we classify the carbons, the number of carbons in the skeleton, and the placement of the carbonyl group. So that's going to determine what type it is. So glucose and fructose, they have the same molecular formula, but their carbonyl group is different. Okay. So here's some examples of different monomers of our monosaccharides. So we have glucose and fructose. Another one would be something called galactose, and then ribose is a sugar, deoxyribose is a sugar for DNA. So these are all examples for you. Okay. So let's see if we can write the formula for a monosaccharide that has 3 carbons. So how would we write that? So we know it has 3 carbons. How many hydrogens? We double the amount of hydrogen. How many oxygens would be equal to the carbon? Okay, so there would be the molecular formula for that monosaccharide if it had 3 carbons. So it's always in this ratio where it has basically a 1 to 2 to 1 ratio. Okay. Okay, so we talked about the monosaccharides. Now let's see what happens when we take one monosaccharide and combine it with another monosaccharide. So what type of reaction has to occur to take one monosaccharide, a monomer, and combine it with another monosaccharide, monomer? How do we bring them together? What type of reaction was that? We gotta dehydrate it. We gotta remove the water. Okay, so here is a dehydration reaction. And basically, when we bring together 2 monosaccharides, we get A disaccharide. Hey, that's really easy to remember because di means two. So two sugars together. So we have a dehydration synthesis reaction occurring between two glucose molecules here, two monomers. Move that water and then you bring them together and we end up with a disaccharide. And this particular disaccharide is called maltose. Okay, so maltose is just two glucoses together to form maltose. And they're joined together by a covalent bond. And this particular covalent bond, we call it glycosylated linkage. So glycosylated, you can it's kind of a sugar-related linkage, basically. So glycosylated linkage or covalent bond between the two monosaccharides together form the disaccharide. Here's another example. We have two monosaccharides, glucose and fructose, and we're bringing them together, removing that water molecule, and we end up with sucrose. So sucrose is another name for our table sugar. So if you use sugar in baking, you're basically using a disaccharide that it contains glucose and fructose. Okay. So sucrose is a very common disaccharide. All right. So let's go ahead and take a look at a video to kind of explain this. It's a very quick video.
Carbohydrates include sugars and polymers of sugars. These molecules are used as building materials and sources of energy. Single sugar molecules, called monosaccharides, and linked pairs of sugars, called disaccharides, are important as building blocks and cellular fuels. Polysaccharides are long chains of sugars. A polysaccharide, called cellulose, is a major component of plant cell walls. Plant cells store sugars in the form of a polysaccharide called starch. Animals store a polysaccharide called glycogen. Polysaccharides on cell membranes act as cell identification tags. Simple sugars, or monosaccharides, are the simplest carbohydrates. Monosaccharides vary in size. Their carbon skeletons range from 3 to 7 carbon atoms. They generally have a molecular formula that is some multiple of CH2O. The formula for glucose, for example, is C6H12O6. Monosaccharides are often drawn with linear carbon skeletons, but in water, all monosaccharides that contain more than 3 carbon atoms bend around to form ring structures. The monosaccharide glucose is a major nutrient, central to cellular metabolism. It's broken down for energy in the process of cellular respiration. The carbon skeleton of glucose can also be used to build many other organic molecules, including amino acids and fatty acids. Glyceraldehyde is the monosaccharide that's the energy-storing molecule produced by photosynthesis. 2 glyceraldehyde molecules combine to make glucose. The monosaccharide galactose combines with glucose to form lactose, the disaccharide in milk. Fructose is the monosaccharide that's sometimes called fruit sugar, the one that makes apples and berries sweet. Fructose and glucose combine to make the disaccharide sucrose, or table sugar. Ribose is the monosaccharide that's an important component of RNA and ATP. A modified form is used in building DNA. Sometimes organisms link sugar molecules in pairs, forming disaccharides. Here are several examples. Plants make sucrose by joining glucose and fructose. Sucrose circulates in plant sap, and we obtain it from sugarcane and sugar beets and use it as table sugar. Lactose is formed by joining galactose and glucose. Lactose is the disaccharide that gives milk its sweet taste. Maltose consists of 2 linked glucose molecules. Digestion of starch in a sprouting seed or in the intestine of an animal produces maltose. Polysaccharides are polymers, long chains consisting of hundreds to thousands of linked monosaccharides. Starch represents a compact stockpile of glucose units linked together in a chain that can be stored by a plant for later use. Glycogen is the polysaccharide that stores glucose for energy in animal cells, especially those in the liver and muscle. Glycogen is the animal equivalent of plant starch, but its molecules are more branched than starch. Cellulose is the polysaccharide that plant cell walls are made of. It's the most abundant organic compound on Earth. Like starch, Cellulose is made of glucose, but the chains are made from a different glucose isomer, so they have different properties.
Okay, so that was an overview of carbohydrates in general, but we also saw how the disaccharides are forming. And we'll get into the polysaccharides in a minute, but that gave us the function of not only the glucose and the monosaccharides, but also gave you the different functions of the polysaccharides, starch, glycogen, and cellulose. So let's do a checkpoint question. Lactose, we just saw that in the video, is where we have glucose and galactose formed together to form lactose. So what would be the molecular formula of lactose if glucose and galactose have the same molecular formula of C6H12O6? So what would be the molecular formula of the disaccharide lactose? So sometimes it helps to write it out. Okay, so if we have glucose is C6H12O6. Sorry, I'm kind of doing this with a mouse, so my writing's kind of bad there. And galactose has the same formula, C6H12O6. Okay, so we're going to undergo a reaction to form lactose. So we know lactose is a carbohydrate, it's a disaccharide, so it's going to have that C, the H, and the O. We just need to figure out the numbers there. What also needs to happen when we bring together two monomers, what type of reaction was that? Dehydration reaction, right? So what's going to be removed? A water molecule. Okay, so that might give you a hint of how we're going to solve that. So we have to remove that water. Let's go ahead and start with the carbons. How many carbons? We're going to have 6 plus 6 or 12 carbons. How many hydrogens? 12 plus 12, 24 minus 2. And how many oxygens? 12 minus 1, right? So the formula, the molecular formula for lactose is C12H22O11. Okay? All right, sorry about that. Okay, so let's get into the polymers. The polymers would be the polysaccharides. And like the video explained, the polysaccharides are starch, glycogen, and cellulose. So starch and cellulose are part of plants. Glycogen is part of animals. So, and they all have a similar function. Well, at least starch and glycogen do. They are storage for energy, because we talked about how the monosaccharide glucose is the primary energy source for our cells. And it's also an important building block for other molecules. So the starch is basically just units, subunits of glucose that are stored inside the plant so that it has a regular energy source. Animals, we also store glucose, but instead of storing it as starch, we store it as glycogen, and we store that in our liver as well as our muscles. Okay. And you can see there's slight differences between the structures of glycogen and starch. So the starch molecule is going to be a little bit less branched, but it's still made of the same glucose monomer. The glycogen will be more branched, but same glucose monomer. And then the cellulose is also the same glucose monomers, but it's in a more structured sequence where we have these hydrogen bonds. So it makes it stronger. And so cellulose is not necessarily for storage, but it's for the structure of the plant cell wall. So the cell wall is made of cellulose, these branched glucose bonded together to form this kind of network. And so that's going to add to the strength of the cell wall, right? So those are our polysaccharides. All right, so and like the video said, the cellulose is the most abundant of our organic compounds on Earth because they're found in every single cell wall of a plant. And again, it's just those monomers linking them together. So glucose is very, very common molecule. We actually don't have the enzymes to hydrolyze this glucose configuration. So when you're eating vegetables and leaves and plants and fruit and all that, you're getting a lot of this cellulose, but we can't break it down. And so this is what is known as dietary fiber. Okay, so this is going to help us slow down digestion because we don't have the ability to break down this cellulose into a usable form. Okay. Now, animals, we don't make cellulose, but some animals, such as our insects and our crustaceans, they have this hard exoskeleton, which is made of something called chitin or chitin, and that's very similar in structure to cellulose. Okay. Fungus also has chitin or chitin instead of cellulose as well. Okay, so let's move on. So here's just showing the difference between the starch versus the cellulose. So two things that are found inside the plants. And you'll notice there's just a slight difference in the arrangement of those glucose monomers. So in the starch, you can see where those hydroxyl groups are. They're all on one side. Whereas in the cellulose, they are on opposite sides. And because of that, that's going to change the structure. Okay, so the glucose rings in starch, that's used as just like an energy source for the plant, and it's held together pretty tightly. Whereas the cellulose fibers made of these glucose molecules with the opposing branches, they are going to build the cell walls. So that's just going back to the structure equals the function. So slight changes in structure affect the function. Right. So what are the functions of those carbohydrates? We saw the video talked a lot about that. So for one thing, it's an energy source, right? Glucose is our main energy source for our cells. Our cells break it down in order to get ATP, otherwise known as cellular energy. We store that energy in the form of polysaccharides, glycogen for animals, starch for the plants. It can also be used for building structures. So remember how the video talked about how glucose, once it's broken down into glucose, that can be used for making, be combined with other molecules to form other types of macromolecules. So there's a structural component to it. So lots of things like amino acids and fatty acids might contain the breakdown of those glucose molecules. Also, think of cellulose. Cellulose, we can't digest it. It doesn't provide any energy source for us, but it provides structure for those plant cell walls. So energy and structure are those functions of the carbs. All right, so let's get into the second category of macromolecules, the lipids. Now, I always think of lipids as fats. But they're not just fats. There's other things in there too. So you can think of it as, all fats are lipids, but not all lipids are fats, okay? And what's interesting about lipids is that they do not form true polymers, okay? So we don't have, it's not simple like with our carbohydrates where you have the monosaccharides or the monomers and the polysaccharides are the polymers. They don't have true polymers. It's just unique where you have different monomers of different types kind of moving together. The reason why they're classified as lipids, though, is because they have little or no affinity to water. So that means they're hydrophobic. So all lipids are hydrophobic. Okay, so that's one characteristic. And the reason why they're hydrophobic or mostly is because they're mostly made of hydrocarbons. And hydrocarbons are formed through nonpolar covalent bonds. So going back to last week, we talked about hydrophobic versus hydrophilic. So hydrophilic is water-loving, and those molecules tend to have polar covalent bonds. So they can, interact with water, which is a polar covalent bond molecule as well. Hydrophobic, these are non-polar covalent bonds in their hydrocarbons, so slightly different. Okay. Now, one thing that's similar to carbs, though, is that it's a CHO. So remember the Amoeba Sisters video where you have the carbs are CHOs, we have CHO. Lipids are also mainly CHO. Okay, so well, they all contain CHO, I should say. There's other things in there as well. So most biologically important lipids we can classify them as fats, phospholipids, and steroids. So those are the main lipids that we have in our body. So we think of them as fats, lipids, or fats, phospholipids, and steroids. So let's talk about fats. So fats are constructed by basically two monomers, glycerol and fatty acids. So I'll show you in a minute what that looks like. But glycerol is basically just a 3-carbon alcohol. Remember, alcohols, they have the hydroxyl group. So it's 3 carbons with a hydroxyl group attached, basically. So that's a glycerol. And then a fatty acid consists of a carboxyl group. What was a carboxyl group again? A carbonyl and a hydroxyl. It's got that, C that's double bonded to an O and then single bonded to an OH. So that would be attached to a long carbon skeleton. So we have the glycerol that has the functional group hydroxyl attached on one side, and then we have the fatty acid that has the carboxyl group attached. Okay, so those are those functional groups, remember? Okay. All right. So let's watch an animation on that. Gotta put my pen away.
Lipids. Lipids are a diverse family of molecules that includes fats, phospholipids, waxes, and steroids such as cholesterol. One feature that all lipids share is their low solubility in water, a characteristic that is important to the functions of many lipids. Cells often contain droplets containing fats that are large lipid molecules rich in stored energy. Adipose cells in animals are almost completely filled with fat. In addition to storing energy, adipose tissue also insulates the body and cushions organs. The membranes around and within cells are made mostly of phospholipids. Cholesterol, a type of lipid called a steroid, is also a component of membranes. Butter, lard, margarine, and salad oil are composed of lipids called fats. To make a fat molecule, 3 fatty acids bind to a molecule of glycerol. For this reason, fat molecules are technically called triglycerides. Fat molecules do not mix with water because they have 3 long nonpolar hydrocarbon tails. Cells use fats for energy storage because the tails hold more potential energy than other biological molecules. The fatty acid tails in a fat can vary in length. A very important characteristic of fats is the number of double bonds in the hydrocarbon tails. Fats that contain only single bonds are called saturated fats. Fats containing double bonds in one or more tails are called unsaturated fats. Unsaturated fats tend to be liquids at room temperature. Vegetable oils are examples. Saturated fats like butter and lard are solid at room temperature. Saturated fats in the diet can lead to heart disease, while unsaturated fats are safer. Phospholipids are important components of cell membranes. Like fats, phospholipid molecules contain fatty acid tails linked to a glycerol portion, but in phospholipids, a group containing phosphate replaces one of the tails. This makes the molecule ambivalent with regard to water. Part of it is polar and mixes with water, while part of it is nonpolar and excluded from water. Lipids also include a family of molecules called steroids. All steroids have the same carbon skeleton made of 4 linked rings. They differ in what is attached to the rings. Let's review the function of these steroids. Cholesterol acts as an important component of cell membranes. Estradiol is a primary female sex hormone produced in the ovaries. Testosterone, on the other hand, is the most abundant sex hormone produced in the testes, while vitamin D aids in calcium and phosphate metabolism.
All right. So that was an overview of our lipids. So let's get a closer look at the fatty acids. We saw from the video how the triglycerides basically have the glycerol and the 3 fatty acid chains. So let's see how that happens. Now remember, I told you that the glycerol has an OH group attached, whereas the fatty acid has the carboxyl group attached. So you can see the carboxyl group here, you see the OH group here. And that is going to undergo a dehydration synthesis reaction because we're removing water in order for that fatty acid to unite with the glycerol. Okay, so in this case we have 3 fatty acids attached, so that's called a triglyceride. And where they're attached between that oxygen and that carbon, that is called an ester linkage. So it's a covalent bond, like what we've been talking about with the monomers of carbohydrates uniting with a covalent bond, but that was called a glycosydic linkage. So this, because it's in lipids, it's called an ester linkage. Okay, so now we have our fatty acid chain. Now, this fatty acid chains, these fatty acids, they can vary in length. So it depends on the number of carbons and the number and locations of those double bonds. So from the video, you saw how some of those fatty acid chains, they are hydrated with hydrogen. There's hydrogens all over. There's no double bonds anywhere. And so it has a maximum number of hydrogens possible. So all of those carbons are attached to the maximum number of hydrogen atoms as possible. So no double bonds. So think of it as saturated with hydrogen. So that's a saturated fatty acid. Unsaturated would include 1 or more of those double bonds. And so there's not as many hydrogens. And so it's unsaturated. It has less hydrogens attached because of those double bonds. And we saw the difference between the two. The saturated fatty acids, they're all going to be kind of compacted together. Whereas the unsaturated, the double bond gets like a little kink in it. And that makes it more spaced out. And so that's why our saturated fats tend to be solid at room temperature. Our unsaturated tend to be liquid. Okay, there's more space in between the molecules. And so because of those kinks. So some examples of our saturated fats would be a lot of the animal fats typically are saturated. So think of butter and basically all the good stuff, right? The unsaturated fats are the vegetable oils, so come from plant-based mainly. Okay. Anything else I need? Oh, I should probably talk about trans fats. So you probably know that trans fats are really bad for you. You don't want to have trans fats in your diet. And what a trans fat is basically a way that we take a vegetable fat and we hydrogenate it. So we add hydrogens to it. So we basically take something like this and we add hydrogens where those double bonds are. And so we change it into a saturated fat essentially. But it's a trans fat because we're changing it into something it's not supposed to be. Now, our body has a hard time figuring out what that is. So that's why it's contributed to coronary diseases or, affect your hearts and your blood vessels, because your body doesn't know what it is. It's this weird, looks kind of like a saturated fat, but it's not a saturated fat. It's something else. And so your body just doesn't know how to process it, and so it ends up causing a lot of issues. So that's why diets that are high in trans fats are linked to coronary issues, things like heart disease. Okay? So avoid those trans fats. All right, So let's take a closer look at another type of lipids. So we talked about the fatty acids. We're also going to talk about the phospholipids and the steroids. So the phospholipids are a very unique looking lipid because they have a nonpolar hydrophobic tail and a hydrophilic head. Okay, so remember how lipids are supposed to be all hydrophobic? Well, it's still considered a lipid because it's got the hydrophobic tail, but it has a hydrophilic head to it. And so that's what makes it very unique in structure. And it actually is the component of our cell wall, our cell membranes, excuse me, our plasma membranes. So you can see how we have a phosphate group, so that functional group phosphate, attached to the fatty acid chain. So there's the glycerol with the fatty acids. We only have 2 fatty acids, so it's not a triglyceride. It's a diglyceride. And then we have that phosphate group attached to the other side. And so that's going to make it polar. So we have what we call this phospho head and a lipid tail. So the phospho head is hydrophilic. The hydrophobic ends are the tails. And so our cell membrane is what we call a phospholipid bilayer. So it has 2 layers of these phospholipids. And look at how it's orienting. So we have the phospho head, that's hydrophilic, facing outwards. Here's the other phospho head. It's facing outward. It's hydrophilic. And then the tails are facing inward because they're hydrophobic. And so that's what's creating the boundary of our cell. So our cell is separated from the outside environment by this plasma membrane that's made up of hydrophilic heads and hydrophobic tails. Okay. So that's our phospholipids. So they make up our cell membranes. And then the last one we're going to talk about are our steroids. So steroids are unique in the sense that they have the same kind of carbon skeletons. And these functional groups determine their function as a hormone of some sort or a steroid. So we have, here's an example of cholesterol. So we have these 4 carbon rings, and then look at all these functional groups. So what's that one down there? It's a hydroxyl group. What's that one? A methyl group. Okay, so we have different methyl groups in this hydroxyl group forming this steroid. And cholesterol, it's an important steroid because it's actually a component of our plasma membrane. So let me just go back. This phospholipid will have little cholesterol units in there, and that helps stabilize it. So it's essential in our animals. But we don't want too much because then we have this steroid hanging out in our bloodstream. If we don't, if we eat too much cholesterol in our diet, it can start just hanging out because it's not being used. And then that can contribute to arteriosclerosis, or the hardening of our arteries, causing heart disease, leading to perhaps a heart attack. All right, so let's get into the functions. So our functions of our lipids, they are an energy source. Okay, so we usually store them. You saw in the Amoeba Sisters video how if you were going to swim the English Channel, that 21 miles, you need to have a lot of lipids stored in your body as an energy reserve. So they are used for that. They're also protective, so they can be used to protect the different, because of their hydrophobic nature. It protects your skin from absorbing a bunch of water, for example, because you have this hydrophobic cell component. So oils tend to repel water. So think of like a duck floating in water. The water just kind of recedes off of its feathers. It doesn't get absorbed. So it's keeping that duck afloat, but it's also keeping the duck nice and warm, and inside it's not going to get all wet. It also is protective in the sense that All of our organs have a layer of fat surrounding them. So when you do jumping jacks, for example, you don't want your liver jumping, banging into your heart or something. That would hurt. And so it's a protective source. It kind of creates a cushion. And then we also know the structural component because of those phospholipids make up our cell membranes. Right? So those are the functions. All right, so here's a couple questions for you. All lipids, A, are made from glycerol and fatty acids. B, contain nitrogen. C, have low energy content. D, are acidic when mixed with water. Or E, are poorly water soluble. So what's the correct answer? What unites all lipids or categorizes them? It's their hydrophobic nature, right? All right, so we're going to stop right here. We went through carbs and lipids. We'll hit the proteins and the nucleic acids in the next lecture. So I'll see you then.